Okay, I'm going to be ready to go whenever I hear the go. Ah, that's the go sign. So yeah, welcome everybody. Thanks again for inviting me. I'm going to be talking, as Daniel indicated, about something called mgRAST. Some of you may have heard about it. Those of you who haven't, I'll try to educate you at least in some ways about what it is that we do. We are probably the biggest resource right now that will analyze and also act as a portal for metagenomics. A bunch of users doing that, and over the past couple of years we've been able to sort of learn quite a lot interacting with our users, and I'll try to walk you through that, and I'm hoping this will serve as a good introduction. I'll try to be provocative in many areas, and then hopefully be able to highlight those things where I'm trying to be provocative, and you'll find them. So if I had given this talk a while back, metagenomics would have been a term that would have probably gone without an introduction. These days I find that we have to introduce it because the term was somewhat watered down with the Human Microbiome Project. And where's Mihai? He's partially to blame. He was part of that, admitted, but so was I. The term got watered down because people started using the term, where's the laser pointer? People started to use the term metagenomics also for what I like to term amplicon studies, using 16S, 18S, or ITS, or other amplified regions. So single gene reporters for an entire chromosome, and as most of you probably heard about, there's an entire industry that has evolved around that. The names Chime, Mother, and some other tools may ring a bell. So this presentation is not about those. So if that's what you're interested in, I have to disappoint you, and you probably just won't leave, because I will be talking about shotgun metagenomics. So random shotgun DNA sequencing of data of samples from the environment. So as I know there's at least one soil scientist in the room, I'm going to be provocative. You take a piece of dirt. They don't like it, of course. Then you sieve it, you apply a kit to isolate the DNA, and here one of the problems already starts because there's multiple kits on the market, and we've learned recently that the choice of toolkits here, of kits, actually determines to a very large extent the representation of the DNA you obtain once you apply the kit. And there's a publication by Tim Vogel's group from a couple of years ago, I believe it's an AEM, where he shows that the signal that you can, or the bias that you obtain from using the kit actually by far outweighs any differences between the samples you might observe. However, then we apply the magic wand, which I guess is why I'm here, because that's genomics and computing, and that lets us in turn answer two questions. Who are they, and what are they doing? Who are they? Something, of course, I can also, at some level, answer by using amplicons. What are they doing? To some extent as well, I'll get to that later. But you really get a better picture, at least in my belief, of the functions contained in the genomes for our specific sample from using shotgun metagenomics. And just to give credit to the people who have been doing this for a while, there is something called the sequencing of environmental clone libraries that has been going on for a very long time, and George is looking at me, because I believe he was involved in that. This is actually much older than any of the technologies I've been talking about so far, but this has been associated closely with Sanger sequencing. Sanger sequencing was around since 1977. Most of the time when I'm talking about, or when people are talking about the rest of these metagenomic studies, we'll be associating them very closely with what is called next generation sequencing. So technology that evolved in the second half of the early 2000s, and that has dramatically changed the cost function. In fact, we probably wouldn't be here if the costs hadn't been altered so dramatically. I think it would be fair to say that in the old days, your university would be providing your computing, or your institute would be providing your computing environment, and you would write grants for the sequencing. These days, I believe in some institutions, it's the other way around. You can get the sequencing for free from your organization, or almost for free, but the computing is where everything breaks down. Okay, oh, actually I can use this. I'm not used to this. Okay, why am I here? Because I operate this thing with quite a few people called mgRAST, which hosts over 100,000 metagenomes, 45 terabases of data roughly. We have about 13,000 users, and these days we process about 1.5 terabases of metagenomic data submitted by users over the web. This gives us a rather unique insight into what's going on. One of the benefits is we usually see new and emerging sequencing technologies and also softwares that is used to prepare the data relatively early. And I'll try to leave you with some of the lessons that we've learned. These are just some of the visualizations that you can produce with mgRAST. I'll get a little bit into what MGRAST is. It actually, like I think many successful projects, started basically as an accident. Rob Edwards and I were sitting together in San Diego on a very nice day on the beach, and we were thinking, what can we do as a team? And we decided metagenomic analysis was it. And we had a very vague idea of what metagenomic analysis meant, but we actually did set up a system that basically allowed the user to upload their data. We provided some normalization, which in the early days was very, very limited. We provided similarity analysis. In the old days, we actually ran BLAST-X to be precise, and actually created a metabolic reconstruction and a community reconstruction. And that was actually surprisingly successful. We had the engineering skills to set up an automated system, and we literally walked away. We had set the system up, there was an automated account registration scheme, and a couple of months later we had hundreds of users, and we really didn't know what happened to us. So without knowing it, we had opened up the floodgates here, because a lot of scientists were trying to do metagenomic analysis and were held back by the lack of tooling. And we actually then went on to create a number of versions. And the version we released in March 2011 is the first one that really allowed analysis of Illumina data. Illumina now is at about 90% of the market share, I believe, for next generation sequencing. So with allowing Illumina, our data volume exploded, and when we hit 10 terabases, we thought that was a big deal. These days, we're well over 40 terabases, and we recognize that this will probably continue, and there's a growing need for the community to sort of face the analysis challenge. Yeah, when I'm saying this, I want to basically mention as a background, there is a paper from a couple of years ago, and I usually forget who the authors are. The title is Sequence Length Matters. And at least one of the vendors will provide you with a t-shirt because they have long reads and they claim that their read length is really important. What I would like to add to this, it's mostly sequence quality that matters more than read length. And let me just show you some examples of what we've encountered. So these are some representations of sequence quality. Let me introduce that example first. And you see here what we call a nucleotide position histogram. Other people have different names. We are plotting the relative frequency of each of the bases across the 100 base pair read. And you can see here that this seems to be shotgun metagenomics because the representation of all the 4 nucleotides in each position is relatively random, and is 25%. So to at least for my expectation, this is what I would expect to see in metagenomic data sets. I see some people nodding in the audience, so we've seen this before. If you've seen a lot of these, you'll also notice the little squiggly bits at the start of the sequencing. It seems to be that every sequencing platform leaves its own fingerprint. This happens to be the Illumina fingerprint, at least for a specific chemistry version, but there's always a little bit of confusion at the beginning of the read. It used to be in Sanger days that we would see sort of artifacts at the end. These days we see them at both ends. But then in any other case, so this looks relatively normal. There doesn't seem to be anything wrong with the data set. And we have a second indicator here that is the duplicate read inferred sequence error estimation, or called DRESI, also called DRESI, invented by Kevin and Will from my group. And this is actually using the artifacts that we obtain in next generation sequencing. What we get in all platforms that I'm aware of that use PCR is we get multiple reads from the same templates. And usually these are filtered out in deduplication. But the interesting thing is if you have multiple reads from the same template, this is not just a problem, it also is an opportunity because you can use it to measure the degree of variation, and we've done that here. And you see that there's very little variation within the reads that come from the same template, and that seems to agree with this as well. We find that DRESI is a much better estimator for our ability to analyze a data set than the FRET scores, for example, because those haven't been designed for next generation sequencing. And so we find that frequently, if the DRESI score is off, we see that something is going on with the sequences. And that could be, for example, something like this. By the way, I was asked by a program manager to introduce these blue bars because nobody likes to see their funded programs sort of produce data like this. This was actually published in Nature. What they've done here is they had 2 problems at once. Not only do they have a barcode problem and an adapter problem as well that led to a signal that looks like amplicon sequencing. It is because it's a barcode, 53 bases usually. And then they also have a poly-A problem because a lot of that seems to be poly-A going on here. So what we are seeing is this is an unsalvageable run. Whatever happened here, the representation of nucleotides is so off from what it would actually be in your sample, that the only thing you can do is toss this. So why am I mentioning this? Because it is probably the single biggest problem. I apologize, I'm a little bit jet lagged. You can actually, you can see these days if you're looking at large quantities of metagenomes. Surprisingly, because we thought that this was something that would actually be solved already by the software provided by the vendor, this is not the case. We see lots of people submitting data sets that are just pathologically broken. In fact, Will here has a presentation that he calls sequence pathology, where he enumerates the various types of problems that are showing up, and we see a lot of that, unfortunately. While I've shown Illumina data, it is important to note that every platform has its issue. In fact, we can fingerprint platforms and even sequencing centers. And no, we will not show that. What is, I think, really important to note is that the variation introduced by the operators is much larger than the variation that exists between the platforms. And I think maybe for this audience, what's really important is that the vast majority of users lack the tools or the expertise to understand what is going on in their sequences, and that the sequencing providers, I'd say that with a grain of salt, but it's probably largely true, they're investing into obscuring any issues that may exist with their sequences. So it's not in the interest of most sequencing providers, especially if you're commercial, to sort of highlight issues that may be going on. And one of the things we get asked a lot is, so should my student be doing the QC before I submit the data? Because my student has to work with the data, right? And this looks very scientific. The answer is no, probably not. In fact, that raises an issue that exists in this whole metagenomics community, because the analysis becomes a specialized sort of industry almost. And the answer for your student is probably no, because your student will likely do the analysis for what, 1, 2, 3, maybe 5 data sets. You can do a lot if you've done that, but we've probably seen 10,000 similar data sets at that time and have a slightly better idea what is going on with the quality control. And that means, at some extent, we have to change how we as a community approach handling these data sets and how we train people on the use of these data sets. The risk here is that if it's only the portals that do the analysis, then the portals will become basically a black box to the community, and that's certainly not something that we would like to see. But we need to recognize that, you know, applying sort of naive approaches to quality control is not really helping. And we've seen a lot of data sets where this was missed. In fact, we have a sort of a thing in our tutorials we give for MG-RAST, where we ask people to look at their own data sets, and quite a few of them were quite surprised by artifacts like these. And we found one data set that actually holds the current world record, because they decided for some reason to do a 63 cycle Illumina metagenome, and they managed to concatenate 2 31 base pair adapters to each other. So it's a 1 base pair metagenome, and you cannot compete with that, because you can't get shorter than 1 base pair. And a good exercise for students is, what is the science you can still do with 1 base pair? But that's not actually as funny as it sounds when you see the person that has worked with the data for a year almost, you know, trying to perform some analysis and then being unable to. So that gives a little bit on sequence quality. That's probably not very controversial. I'll try to be a lot more controversial when I talk about sequence assembly. If I ask the audience right now, who believes you need to assemble data prior to analysis for metagenomics? Can I ask for a show of hands? Who believes in assembly? Awesome. Okay, let me check the opposite. Who believes we should not assemble? Wow, that is almost 50-50. I'm impressed. Usually this works differently. Usually everybody tells me, oh, you have to assemble. In fact, one of the most frequently asked questions on our help desk is, you know, how should I assemble my metadata, my metagenome, I'm sorry. Should also assemble your metadata, but that's a different story. How should I assemble my metagenome prior to submission? And our answer is usually, if you want to assemble, please do also submit the raw data, because assembly will introduce issues. And I'm going to highlight 2 issues here, and I picked 2 very provoking things. These are actual real-life data, not simulated. And the first one is actually something from a host microbiome sample. It's 1 physical sample, 2 libraries were generated, 2 sequencing runs are associated, and the results are somewhat unexpected. At least they changed a lot of people's view of how this happens. And the second one is even more crass, and I'll walk you through that at a little more length. So the first one is from a well-known microbiome data set. Again, I'm not pointing you directly to it. We're looking at 2 by 100 paired-end Illumina sequencing, roughly 10 gigabases per. We unfortunately don't know the insert sizes, and we don't know whether it was amplified or not. What I'm showing in red is the assembled, that is the clustered, that is non-assembled reconstruction. This is a rank abundance curve. And in blue, I'm showing the results for the assembled contigs and the reconstruction of the rank abundance curve. And the only thing I really want to point out here is that the model that most people in the community would have is, if it is abundant, that is, if it's on the left-hand side of the rank abundance curve, it'll assemble. And what I'm trying to show here, maybe not always. And I shall point out that I spent about 2.5 minutes finding an example. It took me longer to plot this than to find the examples. And I have since found many hundreds of examples where this is true. So we have an assembly actually performed by professionals in this case. And we have the raw data that were merely clustered, and the phylogenetic reconstruction looks vastly different. I'm not saying either one is good or bad. I'm only saying they're vastly different. And also my instinct tells me, I'm not showing that here, but my instinct tells me every program that you apply for assembly will leave a slightly different fingerprint. And as far as I can tell from the program, we'll hear a lot more about assembly, so I'll leave it at that and walk on to my next example, which is, I believe, even more crass. So this is actually my own data. This is from a DOE relevant sample. This is a subsurface community. It is probably one of the earliest Illumina experiments, and we've been learning a lot from this Illumina experiments in metagenomics. We've been learning a lot from this. Done this very early on when the 75 base pair reads were just becoming possible. We used a full instrument run at the time that was 9 gigabases. Nowadays this is less than a lane. We were interested in this site because there is a uranium contamination that actually seeps into the groundwater, and the goal for people handling the field site is to sort of keep the uranium mineralized, which immobilizes it in the sediment. And we were just basically playing with this new technology. So we used unassembled Illumina data, and the goal was to determine the alpha diversity and also look at the protein complement, and only later on were we going to do assembly. So we applied 6NS sequencing as well using the V4 tags, and we saw that there's moderate diversity going on, sort of 500 OTUs, 97% identity. This is using RDP. We've also done the same thing using CHIME. The denoising was also performed, so the results look actually very much the same from the two tools. And we found that, interestingly, there's proteobacteria dominate, and there's a single genus, sulfuricurvum, that is almost 50% of our sample. Very interesting. So considering the last point of our research agenda, this was very promising, because 50% of one sample, that means we likely will be able to assemble something. So next we went on and did the shotgun analysis using MGRAST, and we found, interestingly, a 10% discrepancy between the representation of epsilon proteobacteria. There at the time was no sulfuricurvum representative in the database. This has since changed. My friend Nikos Kopidis at JGI and with Hans-Peter Klenk, they've sequenced the sulfuricurvum genome. But at the time, 37% still indicated, wow, we can probably assemble that. Long story cut short, we took almost 3.5 gigabases, which ends up being 45 million reads, thinking the genome size should be about 3 megabases. And then we ran a simulator, simple Landau-Waterman analysis from the 80s, let me point that out. And it tells us if it's one genome in there, we'll expect an 1100x coverage, which basically means, wow, this is wonderful, this should be really good. Those of you who've played with Landau-Waterman analysis and genome assembly will know that this never works as advertised, but it's a good indicator. It gives you an idea. And then we actually ran Velvet, and then we saw, oh wow, we're actually getting a lot of contigs, but they're very short. And worse, blast analysis showed us there were no epsilon proteobacteria in there. Oops, what had happened? Something went wrong. So okay, so we looked at sequence quality. Was that the issue? And we used the Drisi score that I mentioned earlier, and we also used the recruiting of mate pairs against the contigs, and we found that there was no sequence quality issue. Then we started, since we were computational folks, so my background is computer science, although I've dabbled in genomics for a couple of years now, we thought that we'd done something wrong, so we not only ran Velvet, we ran a whole bunch of tools, and what really makes Velvet a very nice tool is it has very few parameters, or it had very few parameters. So we started working with the tools, and they were all showing some interesting issues, and we learned a lot because 45 million reads for all these tools was a lot, and we saw a lot of technical issues that I'm not going to bore you with. An interesting quote from Mihai, who's in the audience. I use that a lot, Mihai. That was in St. Louis in 2010. You said metagenomes can't be assembled, and you also said all assemblers are equally bad, and I completely agree. You also went on to say, here's my new assembler, which I thought was very funny at the time. But I'm not so sure that is true anymore. But anyway, so we played with all of these, and we found that everyone had their little quirks. Interestingly enough, what we found in this particular data set, what happened is if we change the K parameter, the K-mer length parameter, so for the non-genomics types, in assembly we're trying to puzzle together sequence reads by identifying overlaps. So the K in Velvet is the length of the overlap that is required to basically merge two reads. Okay. So by changing the K, something very interesting happened. So here we're plotting the results. We're plotting the number of contigs and the N50. The N50 is, think of that as a representation for the length of contigs. Okay? So what you're seeing is, as you increase your K here, your length of contigs goes up quite a bit, but your number of contigs goes down. So at the time, I showed this to quite a few people. I believe I even showed it to you, Mihai. Maybe not. I'm not 100%. So I didn't understand that at the time. I'm revealing my own ignorance here, and I should have. Is the only thing I can say, because what we're looking at here is the result of diversity, non-clonal entities. Turns out all these people writing these assemblers had assumed nature was clonal. It isn't. Simple as that. There's strain variation around us all the time, and assemblers at that time couldn't handle that. That was pretty shocking to me. The most shocking thing is, if you think of something like meta-velvet, which is a wrapper around velvet, it'll also fall victim to this. This is quite problematic. So what some tools are trying, they're trying to play with different K values at the expense of runtime. That'll help you some, but we were quite shocked by seeing this. And we actually, basically what we were seeing is, we were seeing too much coverage, so we had to rely on subsampling in order to get rid of this. And you know what's happening here? My hidden slides are being shown because my export to PDF is screwing me up. So I'm going to have to walk you quickly through the hidden slides. So that's the actual representation on the Lander-Waterman plot that we see, and we can do the jackknife with this, but we haven't at this time. But what you're seeing here is that the number of reads is plotted against the number of contigs, and if you've seen an old style Sanger genome, you were seeing this curve go up and then go down and then stay down. What you're seeing here is it goes up again because the newly introduced strain variations basically break the consensus for the assembly. And Nubler, by the way, would have been a very good choice here, but Nubler couldn't handle the data at the point because Nubler just uses a different representation for the consensus. But anyway, we find that even the modern tools at the time, this is a couple of years ago, will succumb to too much strain variation, very interestingly. So strain variation is not something that the authors of those tools were particularly prepared for. Luckily, we were able to build a pipeline that relies on subsampling, and we were able to publish the genome just this year, which shows you that it can be done from a metagenome. This is not the first genome to be extracted from a metagenome, but we've validated this, and it is probably, it may be chimeric to some extent, but whenever we did Sanger sequencing and PCR to validate individual regions, we found 100% match, so we're pretty proud of this one. By the way, interestingly, submitting this, if you want to obtain a real species name, a real name, you can't, because in order to do that, you have to submit 2 clonal entities to the sequence, to the archives, and you can't do that because this is not something you can actually culture. We tried a lot. So there's a cultural problem. You can't obtain valid names, and George can tell you all about that for something that you can't culture. Interesting. And I'm being very provocative here. So the state of the art is we apply a single algorithm, assembly or clustering, and certainly MGRAS is guilty of that because we apply clustering only, no assembly. We assume optimal results, and then we proceed. And that's very tempting. And maybe it's not as provocative as I'd like it to be, because everybody is sort of confronted with these really large data sets. A single lane of Illumina is 10 gigabase pairs. That easily is 20 gigabytes on disk, and biologists have a hard time shipping these files around, handling them, viewing them. So what this does, it very quickly reduces your metagenome to a very manageable size. Of course, there's also huge bias introduced. So in many ways, I believe the community has to overcome this. So an alternative, there was a couple of papers that were published last year. Actually, I also published this year. So there is something called non-parallel from Konstas Konstantinidis' group at Georgia Tech. And we've published something called the K-mer spectrum analyzer. What these are doing is we're trying to understand coverage strata, and I'm introducing the term and I'll define it in the next slides. So we don't use subsampling as was done in non-parallel. We use a K-mer index, and we come up with a classification of basically areas in the metagenome, strata in the metagenome that exist at different coverage. And the reason for doing that, in my mind, the rationale is you can apply one algorithm for this stuff that is between 1x and 3x coverage. So sequence assembly for that, this is a soil metagenome, very typical, it's very diverse. If you try to sequence assemble this, the expectation would be you wouldn't see very good results, right? Because there's very little overlap. And you probably need a different strategy for this area. And the stuff that is between 3x and 10x, you could even still debate, is it worth sequence assembling? If you run sequence assembly on next generation data, I think typically your coverage is upwards of 40x with short reads. Here, assembly, if applied, what it would do is give you very few contigs. They will likely not be longer than your average read size. And the big question is, how reliable are they? So clearly you want to use a different approach for this metagenome than you'd use, for example, for this. This is a microbiome sample. And here you can see that a lot of these have very low coverage, but more and more we find reads that actually have very high coverage. So everything to the right of this, what's that, light blue line exists at more than 30x. So clearly you can run sequence assembly and expect good results, right? So we're hoping that this approach, which we also applied for our subsurface sample, and this is our genome of interest, but you can see there are other genomes out there, shouldn't say genomes, replicons out there that exist at much lower coverage. And had we applied at the time an approach like this, we would have seen that we needed different strategies for each of the different strata in the metagenome, allowing us to see things a lot more clearly. So maybe not so provocative. Was that depressing enough for metagenome analysis? Because if not, I'll be a little more depressing. A couple of years ago, Will from my group, he actually published let's just say a method survey looking at gene prediction tools. And again, the gene prediction tools suffer from, I will say, fundamentally the same flaw that the assembly tools suffered from, because the world is clonal and it's also noise-free. It's actually also kind of interesting to determine what is noise and what is non-clonality, because I believe the only approach is sufficient coverage and use of statistics. So here what we've done, we've used established data sets that were being used in the comparison literature that had a known error rate, and they're actually much longer than they're longer still than the average read length that we are getting these days. And we applied a number of tools, FracGeneScan, MGA, MGM Prodigal, and Ophelia, and we looked at the performance of these tools with increased error rate. And I think the only thing I need to point out is that for all but one tool, the recall rate here is below 30%. So we're missing 70% of all genes if there is a 2.5 to 3% error rate in the data, which unfortunately is quite reasonable as an assumption. So the outlier here is FracGeneScan, because believe it or not, the authors had anticipated that sequence data isn't always perfect, and they've included a model for errors in their underlying representation that allowed them to perform much better than this. I think the take-home message here is that the method community needs to embrace the notion of data being flawed and introduce measures of noise in sequence and then decide which tools to apply. We're happily running FracGeneScan and MGRAST, but I was surprised to see the level of performance of these other tools. By the way, this is probably a bad estimator of overall performance, because these are Sanger reads and the error model is also using the Sanger error model. I believe reality is probably a little bit worse than what we're seeing here. Okay, so let's assume we've done all the steps so far and we want to run a functional annotation. Let's do a very simple experiment. Can I recognize samples from the same environment, so from human microbiome or feces or soil or maybe from a bioreactor? And if I use metagenomics to look at all of these, will I still be able to see the differences? So here's what I'm doing. I'm using seed subsystem level 3 functions reconstructed by the MGRAST pipeline. and I'm seeing that, you know, the what was blue again? The feces cluster differently from the human microbiome, from the bioreactor, and also very nicely from the soil. They're all separated. So wonderful, metagenomics works. There has been an article out there saying metagenomics doesn't necessarily work. They were using amplicon metagenomics, and their metrics were a little bit off, but yeah, it works, wonderful. Let's now do the same thing, but let's not use seed, let's use KEGG. Seed subsystems, many of you have heard about that. KEGG is of course a much more established way of looking at metabolic pathways, and suddenly the picture is much less clear. So simple change in the pipeline. Mind you, I haven't changed the gene calling, I haven't changed any parameter at all. The only thing I did change was the way we reconstructed from what we call, we mapped from the profiles, which are an internal construct, an internal model of the metagenome, how we projected to seed profiles or KEGG profiles. This is just another way of writing down the same results. Okay, and that already introduced a significant bias. It may have to do with a different level of representation. So how many of the sequences in our metagenome were we able to describe using the various ontologies, but that's just a one idea. And then it needs to be pointed out that we typically pick subsystems because they have the best coverage. So they best cover your metagenome. Whether they best describe it or not is obviously something that needs to be discussed. Okay, that was the depressing part. Anybody still wanting to use metagenomics? I hope. So I'll walk on to some more hopefully exciting and some more recent work. So what I've been mentioning is that we are seeing a lot of the data. This is actually the throughput on a monthly basis of MGRAST, which you can see is well over a terabase per month now. And we recently started working in the context of the DOE Systems Biology Knowledge Base. And I'm going to walk you through some of the changes and new developments that are happening with MGRAST and also in the context of the DOE Knowledge Base project. So first of all, and this is maybe one of the most important issues we've worked on in the past couple of years is we noticed that we were spending an inordinate amount of time writing code that had to do with the platform we were using. We were using a very traditional cluster at the time. And we also noticed that, we were limited to one computational system, to one cluster. And we needed a lot of horsepower, so that was limiting to us. So we ended up building a different platform, if you will, for doing computes in the next generation sequencing, whatever you want to call it, era. We actually have a data storage platform, a data management platform called SHOC, which is not a file system, so it isn't like network file system. It's not a database, so it isn't like Oracle or MySQL, and it's also not like MongoDB. It's not a SHOC. It's not a NoSQL database. It's sort of a hybrid of all of the above. What it allows you to do is present arbitrary subsets of the data to a number of computational resources, and they can be basically everywhere. They can be inside your cluster or outside your immediate domain. You might, for example, need a machine with lots of memory or lots of disks to perform a specific task, and the system will make the data available across the internet using a simple REST interface that basically works across all administrative boundaries that we've encountered so far. We also created a workflow engine that lets us easily implement pipelines on top of this platform. And we've used this, I should also quickly explain the analysis task we're going for, and there's a slide missing here, I'm sorry. I should say that we've used this not only in our own machines, we've actually used this on EC2 and with a number of partners, and our throughput has been over 100 gigabases a day. But the system scales very nicely, so by just adding more computational devices to this ecosystem, we can actually increase the throughput almost arbitrarily. And what I find most interesting is not only have we been able to use this, we've had third-party entities basically taking some of our virtual machine images and running them on their infrastructure, thus contributing to the throughput of the metagenomic analysis using their machines and then feeding the data back into the SHOC platform, basically using that to accelerate their computations. So in K-Base, the goal is to build a very fast pipeline that performs taxonomic assignment and functional assignment, and the emphasis here is on the term at scale. So we don't want to do this as an experiment, we want to do this as a capability that scales and that everybody can use as a function. So in the end, K-Base is trying to become a platform that you can use. You can invoke a service, for example, analyze my shotgun metagenome. And you can do that for a single sample, obtaining taxonomy, rarefaction, and function information, or K-mer profiles, or even just using the QC tools. But you can also use it to obtain information on many samples. Obtaining information on functions and taxonomy. One of the important aspects that a platform allows you to do, you can very easily do something like, give me a subset of the data. I would like to see all actinobacteria, or I would like to see all reads from actinobacteria, obviously, in my sample, or I would like to see all the sequences that I can link to a specific metabolic process, say lysine biosynthesis, and extract them and perform my analysis on them. And this is all enabled by the platform. One thing the platform does, it enables you to handle data at large scale. So here we're looking at a comparative analysis using PCOA, and again, seed subsystem level 3 functions, and we're looking at over 2,000 soil metagenomic data sets that we've normalized and then analyzed for subsystem abundance, as I said. And importantly, we are also including metadata. So metadata as defined by the Genomic Standards Consortium, and having this metadata enables us to paint these various reconstructions in whatever metadata we have available. I believe one of them is technology. This is technology, and I believe this is the country the samples originated from. I may be confusing the two. But they're just here as a placeholder, trying to illustrate the value of having metadata. Okay, this is the other slide I was mentioning earlier. So as I said, we have the ability to have third parties contributing computing cycles to the analysis of the metagenomic data. That is, I believe, a new thing. And then we've had the ability to scale the data rather fast. And let me say that again, since KBase is not a resource provider for computing cycles, the only thing that KBase brings to the table is the software platform. It's important that we can use non-KBase computers in this analysis. Again, this slide should have gone. We provide a unified API that lets you use everything in KBase and also in MGRAS. You can use it with REST and JSON RPC. You can use this with multiple levels of access. You can go down to the raw data, or you can use a very high-level function, say, annotate my genome. And we've demoed recently in DC that we were actually able to perform a genome annotation for a microbial genome in under 3 minutes. And if you're willing to wait 5 more minutes, you can actually obtain from KBase an automated metabolic reconstruction, which is kind of a unique feature. We are, for the MGRAS project, we're rebuilding all of our infrastructure around this API currently, and what is maybe one of the more important take-home messages for me as an engineer of this platform, this is completely liberating when you design something. Open this up totally is a completely different environment to work in, and it's a very good experience. Okie doke. The next thing, we also wanted to give people the tools that they want to work with. So in this context, we've made an R language client available. It's called Matter, and it allows you to do representations like this. This is looking at 28,000 shotgun metagenomes, normalized for subsystem level 3 abundance. And before somebody asks, the red is just a presentation artifact. The rendering library uses that. And I need to point out that this looks like a bird of prey. At least I'm being told that. We don't know why it looks like that, but it would be an interesting question for somebody to sort of start looking at that. And also, if you submit a new metagenome, an interesting question that I can imagine is, where in this cloud is my metagenome? What is the most similar sample in terms of sequences, but also what is the most similar sample in terms of metadata? So is it the case that all samples from, I don't know, Urbana-Champaign that were done in the Miscanthus project for soil, they are down here? We don't know that. Would be very interesting to learn these things. So the last thing I'm going to talk about is very much a work in progress thing, and it's also a very ambitious thing here, because what we're actually trying to do is we're trying to create flux balanced metabolic models for metagenomic data, which we can do now using KBase. We're very proud of that. But we also realize that there's a vast set of methods that we can use, and the uncertainty, at least in my mind, in what method should I use is actually significant, and I believe it's much worse for the users because they're really overwhelmed most of the time. So when I'm presenting this, bear in mind this is very much work in progress. So there's a number of methods to go from reads to taxa or from reads to functions, and I've mentioned the MGRAS pipeline as one of them. We can also run assembly. There's a vast set of assembly tools. Velvet is maybe the most widely used one, I believe. Kiki is our own assembler that runs on a big supercomputer, handles up to 20 terabytes of 20 terabases of data, I'm sorry. IDBA UD, for example, is something that is Coming out of Hong Kong, people are mentioning that it's very useful for high strain diversity situations. You know, we can do that. We can go from assembled contigs and singletons, or we can exclude the singletons. All these are possible variations of the pipeline. Or we can use clustering, which is what we normally do, to come up with functional genes. So these are possible choices from a menu, and in fact, there are actually quite a few of them. The other choice is to, if you're going to go for taxa and you don't want to derive it from the functional genes, you can derive it from the ribosomal genes. But even there's many choices, right? Am I going to use assembled contigs? Well, see above. Again, am I going to use singletons or include singletons or not? Am I going to use clustering? Or am I going to run something like emerge that attempts to reconstruct 16S full-length genes from the entire data set? All of these are possible. And once I have that, I can go to models. But when I have those models, then the big question is, we have a pipeline that we know that takes us from functions, takes us to the functions for the observed genes. But the next thing is we can also infer the functions using the 16S proxy gene, using something that came out from Curtis Hattenhauer's group called PyCrust. So PyCrust will infer a metabolic model for a single gene reconstruct for a single gene. Okay, and once that is there, we can then use model seed. This is Chris Henry's work to come up with a full metabolic models and also fill the gaps. I should point out that all of this happens inside KBase. Many of these steps are rather lengthy, so you need a computational environment. Actually, you need to have multiple machines working on that, and that the KBase is ideal for this type of work. So our scheme here is we're using whole genome shotgun, we're assembling, we're clustering, we're using emerge to extract the taxonomic information. We're also extracting taxonomic information from the clusters and the assembly, running into PyClust to come up with functional profiles. In the end, everything results in functional profiles, and as I said, we've included emerge from the Banfield lab and PyCrust from Curtis Hattenhauer's group, and then we're coming up with metabolic models, and then we do comparisons. Why am I showing this? Because there's a lot of variation at every step of the pipeline. And I'm not saying either one is right or anything is good or bad. All I'm saying is just take a look at the number of 16S's. This is all the same sample that different strategies for reconstruction give you. If you were a user of metagenomics, a bench biologist, I can totally understand why you would be overwhelmed. Okay. And this is a simplification because there are actually a lot of different clustering, different assembly algorithms, even different clustering algorithms. So I believe it is probably true that Kiki in many cases has the highest number of anything. It's very aggressive in assembling, and I'm saying that in a positive way. It's not negative. KHmer actually is a very interesting tool that has come out of, okay, my jet lag is killing me. Titus, of course, Titus, yeah. Charlie Brown. Charlie Titus Brown. He likes that. Yes, so that, for example, there were runtime issues with that. But I mean, I'm just pointing this out. We don't know whether anyone is good or bad or right or wrong here. All I'm saying is the uncertainty is quite high. And if you look at the number of OTUs that were reconstructed, that's also a quite striking difference. It goes from 15,000 down to 4. One of them is probably more right than the other, but we can't really tell at this time who is who. And then the good news in KBase, we can automatically create what is called mixed back metabolic models, and there's a graphical user interface for that. We can even perform the gap filling again. There's a nice there's a nice user interface. We have these user interfaces for almost everything in KBase, and at the end result, we get this very nice representation. Here we compare two reconstructions. And we can already see that the fatty acid degradation here in the KEGG overview was reconstructed by one of the strategies and was not reconstructed by the other. I should point out that consensus is light blue, and then stuff only found in one sample is either blue or green. So you can see even in the same sample there's dramatic differences, and since this is ongoing work, I'm not going to dive any deeper. But the good news, since this was done in KBase, we can automate this and run this across many, many data sets, and you can already see there's significant variation in the various tools that we have available here. I should also thank Steve Allison from Princeton for making his soil data available. So this is again soil, very diverse environment, very hard to work with. We probably should have picked a less ambitious example for the first iteration of this. And with that, almost in 45 minutes, which was my goal, I'm done. Thank you very much for your attention. And these are my collaborators. I should point out the persons in blue are the major contributors for the work that I've shown. Thank you.
Thank you very much, Volker, for a really cool overview of many of the issues that we'll be facing throughout the week and following weeks. So we've got time for questions.
So in the first assembly problem that you showed, you had these red and blue bars, and that was abundance estimates of taxa.
Oh, sorry, that was a rank abundance curve. Those were abundance estimates of the various taxa. Yeah, I should have.
That was once for 16S and once based on shotgun data?
No, they were both based on shotgun data. It wasn't clear enough. No, they were both based on shotgun data. One was assembled, the other one was not assembled. Let me try and navigate here. Okay. Yeah, the striking thing is how vastly different they are, in my mind.
It was using the same method for taxonomic assignment.
It was all identical. It was the same pipeline. The only variation was one's assembled, the other one not assembled. And this is not the exception. This is actually the norm.
But could it be that your taxonomic assignment method is affected by sequence length, so that it's really bad for short context or for reads? For what you're using?
That is totally possible. I would say that wouldn't it be surprising? Well, maybe you're right. We did try quite a few methods, not including Phylopisia, but we found this to be true. This particular method is actually the MGRAS pipeline, and I don't have an image here for the other results, but there is some variation between the pipelines, but I don't believe it's that dramatic. And the one consistent thing we see is basically that phyla are disappearing, and it's not just that phyla are disappearing at the tail end of the rank abundance curve. They're disappearing everywhere.
You mentioned about the variation, about the sequencing edges. What do you think about the spiking normalization?
That's a frequently asked question. I believe it's probably a good idea if we as a community got our heads around something like that. So if we agreed on a common standard that we would use for every sample, that would give us some idea of what is going on. The big question is, if you use a clonal entity for your spiking or your simulation, how much are you actually going to be able to infer about your non-clonal mixture about that? So you have to be very careful. I've seen a lot of literature that has been sort of extrapolating from spiking. And they made statements that I find cannot be validated. But yes, this is probably not a bad idea. One of the suggestions in the context of the Earth Microbiome Project was to always, with every run, you would include basically something that is equivalent to spiking in order to normalize between different instrument runs and different platforms. So that could be a very good idea.
There's one PCA problem comparing the seed and the tag.
Mm-hmm.
What are the main reasons?
Can you repeat the question?
Yeah, okay, so the question was, and it's actually, I think it's very similar here. I'll jump on. Sorry for those that are getting nauseous. So there's a discrepancy in the reconstruction between KEGG and seed. And what we find is I can't speak to the latest KEGG database because it's not open source and we cannot use it in our work. So this is an old version of the KEGG database. If you want to use the latest KEGG database, you have to pay, and we are not able to afford the licensing costs for that. Apologize, but I believe what I'm about to say will also hold for the current version. We find that the coverage in the seed, due to the nature of how the analysis is done and how the assignments are being made, is much, much higher because seed is using a controlled vocabulary and is placing a lot of emphasis on consistent annotation, and that allows you to obtain much better metagenomic hits than the other methods, where there's not so much emphasis on the controlled vocabulary. The one observation, and that's not very systematic, but the observation I can make is that the vast majority of the data sets in MGRAS, the seed coverage will be significantly higher than any of the other databases.
Okay, can I ask you about seed?
Mm-hmm.
So you showed us that seed is way more powerful than KEGG in this respect, but is seed itself available? Because
seed is completely open source, and seed is also, the seed platform has also been merged with the K-base platform. So not only is seed available, it's actually more available than it ever was before. One of the shortcomings, I admit, is that currently the download functionality, if you want to use it as a bioinformatics person, leaves something to be desired. That's actually on our goal list for the next phase of KBase. We're working on a much better way to download data products that you would like to use. And actually, I'd welcome after the feedback and ideas on what you would like to be available.
And another question. So you first spent a lot of time convincing us that assembly is not possible and gene calling is not possible. And you described your infrastructure, and then suddenly you were looking at metabolic pathways and so on. So I wonder how confident you can be that you can reconstruct metabolic pathways if all the underlying computations are not possible.
So first of all, my point came across. There's a lot of method uncertainty here. I think we have to face the reality, our users. So bioinformatics as a whole always makes the assumption, and genomics too, that everybody you talk to is a bioinformaticist or a genomicist. We have to face the reality that our consumers are not. They're mostly biologists or environmental scientists or medical researchers interested in what does the microbiome tell us, for lack of a better description. So we have to provide answers to these questions, but we need an error bar. And I think the work I've been showing towards the end is actually approaching this problem. We want to start giving them a level of uncertainty associated with the methods. And you're right, it's very frustrating at this point. Nothing that we do every step of the pipeline adds uncertainty, but that's not unique. Other areas have the same problem. I think what is unique about biology is that we as a community don't have our heads around that. We don't have a standard representation that we can make available to our customers and say, hey, this is the state of the art, and wherever you go to, you'll get something that's very, very similar. Come again in a year from now, and we'll have evolved the state of the art, but there is no consensus. I think that's kind of unique to our field, and there's a lot of funding potential in agreeing on a consensus, in that we would be viewed as good customers and good sort of custodians of the data and the analysis pipelines, but that isn't happening right now. And so I believe what the research that we are showing, or that I was showing at the end, is doing, it's going to help us improve our pipelines. It's also going to add value to what we produce for our users. But yes, educating users as to the level of uncertainty associated with any analysis is, I think, important, and I think the huge variation between the metabolic models is reason for concern. But you also saw that some of them were actually pretty close, and you were able to draw scientific conclusions from that. Right? Fatty acid biosynthesis in one sample was, in one reconstruction, was missing, stuff like that. So it allows you to go down to levels that are actually informative for the end user.
You mentioned the problems with dealing with non-clonal samples, and this causes problems in assembly, also in gene prediction, if I understood you correctly. So I guess people are addressing this question. My question is, how are they doing it, and whether or not things like hidden Markov models have been used?
Funny enough, for the gene calling, using hidden Markov models is the standard approach. Absolutely. I think the big invention of Frac GeneScan is this Eugene Ye's group from Indiana, was the inclusion of an error state, which was an error state. So they use a Markov model representation that not necessarily assumes all three base pairs to be correct. This is probably happening in other areas too. I'm not really seeing a whole lot of that, because I believe it is very difficult if you don't have access to many samples to start writing code that actually addresses real-world problems. So one of the goals for the MGRAS platform that we have is making a lot of data available in an easy-to-use format for bioinformaticians, so that this trend that I'm observing can be stopped, because I'm actually reviewing a lot of papers using providing bioinformatics methods for Sanger sequencing, and I'm sure that's true for most of the people in this room. I just reviewed one the other week, and I was surprised, because in the US it's very hard to find Sanger sequencing, and certainly not Sanger sequencing at scale. But yet what happens is, because what is probably true is that our archives are no longer as functional as we would like to have them, and that may be different again in the EU than it is over in the US. But in the US it's very hard to get your samples into the archives. So a lot of the complete microbial genomes, I believe we have way over 20,000 microbial complete genomes that are not in GenBank. Ditto metagenomes, right? So it's very hard for most researchers to gain access to real-world data that they can write code against. So one of the goals for MGRAS is to act as a platform that provides access for that. I'm seeing some of that happening, but the vast majority of method publications addresses legacy data at this point. Does that answer your question?
Yeah.
Part of this I can't. It's not really a question, but now I can turn it into one at the end by saying, "What do you think about this?" So one thing, since you asked, why shouldn't my student do quality control? They should, because you put the word student in there, and they need to. You said it's hard for users to do stuff and get used to working with data. That's the point when they should do it. Once they've done it 4 times, then they can forget about it because they've learned, right? But and also because I always react against that kind of statement. The leave it to us, we've got loads of experience, we'll do it for you point of view is of course quite scary too, and you also made that point.
Yeah,
I wanted to just.
I was provocative.
I didn't want it to go unchallenged, but I'm not disagreeing. I'm sure we're on the same wavelength completely here. Yeah. But along the lines, because it came up at the same slide you were showing, was that can you give a comment on the vast difficulty of knowing whether the artifacts in your data are artifacts in what you sequence, library preparation, PCR, which is sort of part of the library preparation, or the fact that you're rubbish at running your sequencing?
So yes, we can. In fact, we've become pretty good at profiling this. So let me point out that I'm completely in agreement with you. The black box approach is really bad. Some of the people in the audience were part of a class that I used to teach at Bielefeld University, basically demystifying the pipeline. I think it's important that your students are exposed to every transformation we do on the data. Do they actually have to run it? Finally, maybe not, but I'm almost convinced that they have to be familiar with the quality control, the gene calling, and the challenges, because only using that approach you can actually identify what may have gone wrong, and you're not, I've been called by a researcher recently that found something really astonishing. Soil was deemed to be the most complex environment around, and they found repeatedly that it only had 500 OTUs when they did metagenomics. And yeah, Penny, it's kind of funny, but they actually ran MetaVelvet, and that very nicely took their 20 gigabase samples and reduced them to 600 kilobases. And they weren't doing anything wrong. They used the standard recipe for metagenomics, right? So yes, it's very important we do that. Second answer, yes, we have a person in our group that actually spends a significant fraction of their time debugging sequencing runs. We actually, this time, and I'm happy to show you some tools offline. We have visualization tools for Illumina runs. We can show you a little movie of what's happening in your lane, identifying stuff. We can visualize the bubbles, the famed bubbles that are appearing, and to allay some of the fears. Most of the errors are not in running the sequencing instrument. Those are very rare these days. It's mostly library preparation. The challenge is library preparation, especially at scale. That is far more difficult than running the instruments. Is that answering your question? No, your comment, okay. You're right, it's not, but the question is, how do we address as a community the student educational part? That is a really big problem.
That's a good question to think about over lunch. So we'll resume at 1:45, and let's thank the speaker once again.